All right, I shared my computer already. So let's start to review. For the final exam, you will have the 100 marks for the exam paper. Include the 50 single choice questions. It's 1 mark each. So you will have the 50 questions. And have the 4 glossary questions. I will give you the 1 item and you will explain what this means, what this glossary means. And you will have the 4 questions, 5 marks each. And the third part is the essay question. You will write the answers with the following the questions as much as possible. And you will have 3 essay questions and 10 marks each. All right, so we have the 3 lectures. We will help you to review only for the 1 time today, and we not repeat anymore. And these lectures of this review will not upload. And for my part, the first part will be for the 30 minutes, and the next is the Dr. Chen's part. It's 30 minutes, and the third part is Dr. Ger's part. It's 30 minutes. Okay, so let's start. Let's review. All of the examination questions is will from your PPT lectures. So you can review your PPT lectures. So for the enzyme, what's the important part for this chapter? Firstly, you have to know some of the glossary, like what is the simple enzyme? It's just the protein part. What is a conjugated enzyme? It includes the Apple enzyme and the cofactor. And the whole enzyme called is the holoenzyme. Here. And the simple enzymes, it's required not other chemical group, just the proteins for its activity. But the conjugated enzymes, it depends on not only the protein part, but also the non-protein constituent. And the cofactor is a chemical group, and Apple enzymes is protein part. Next important part is Michaelis Menten inclusion. Michaelis inclusion, what you have to know. So firstly, you have to know the inclusion. What is inclusion? Just remember and recite it. So it's V0 equal to Vmax times is the substrate concentration and the derivate the Km plus the substrate. And what is each item mean? What is the V0? What is the Vmax? What is substrate concentration? And what is the Km? Km is Michaelis constant. Next, you have to know the glossary of isoenzyme. Isozyme. The difference doesn't mean the different isozymes catalyze the same reaction, but differ in their primary structure and the kinetic properties. And what's the properties for these isozymes? Okay, and in the lectures, we will give you an example. Give you an example here is LDH. There are 5 types of LDH. The LDH is isozymes, and what is the LDH means? And what's the function? What's the function for each LDH? Okay, and then I will give you maybe some questions for the medical application. So this is LDH. It's very important. Next is what is the mechanisms of enzyme action? Firstly, it's a property of enzyme catalyzed reactions. It have 4. It's high efficient, high specific, can be regulated, and non-stable. Next is the catalytic mechanism of enzymes. It's a formation. It's the EX complex, and what is the induced feed model? And it is the induced feed model. Okay, here. 陈老师在吗？陈老师，陈老师在吗？ Okay, so the induced feed model is important. So you have to know what is mechanisms and what is the induced feed model. Next is the reversible inhibition. This is very important. And you have to know we have the three types of reversible inhibition: competitive, non-competitive, and uncompetitive. And you have to know for the everyone, what is the competitive inhibition? And what's the properties for the competitive inhibition? You have to remember and recite it for this part. Okay, so this is the inhibitor compete with the substrate for the binding to the active site of the enzyme. And include this, we have the two concepts. It's AKM, it's a parent KM, and the VMAXI. So you have to know what is the KM, this KM means. Okay. So this is a summary of competitive inhibition. This is all in the lectures. It's all in the lectures. And in this part, we applied medical application is the drug. It's a sulfonamide drug. So you have to know what is the mechanism for sulfonamide drug, because sulfonamide drug is a competitive inhibitor for the enzyme. This enzyme is dihydrofolate synthesis. This enzyme is very important for the bacterial reproduction. So the sulfonamide drugs competitive inhibits the enzyme activity. So it can prevent the bacterial reproduction. Okay. It has the structure similar to the PABA. So next is non-competitive inhibition. And you have to know these slides here. They are from the lecture slides. You can go back for your original lecture slides and to review it. And the uncompetitive inhibition. And this is the properties of the AKM and the Vmax of different inhibitions. So what happens for the AKM and the Vmax in these three types of a reversible inhibition? Okay. So this is the important part for the chapter 3, enzymes. Okay, so next, we'll review the chapter 4. First important is glycolysis. So glycolysis, this is the review for the product of glycolysis. Glycolysis have the 10 steps, and we have the 3 irreversible steps. It's the first step, second step as a 10th step. And the product, if I'm asking the product of a glycolysis, so what's the product? One molecule of glycolysis will produce the 2 molecules of pyruvate, 2 molecules of NADH, and 2 molecules of ATP. And you have to know the regulation in the glycolysis. That means the regulation of the king enzyme. It has the 3 king enzymes. It's corresponding to the 3 irreversible reactions. So firstly, it's hexokinase. Hexokinase is in the first reaction from the D-glucose to the glucose 6-phosphate. And you have to remember the properties of glucokinase. and the physiological roles. Next is PFK1. It's phosphofructose kinase 1. You only need to remember the abbreviation here. You don't have to remember the full name for this one. The PFK1 is the most important. I repeat again, it's the most important the regulatory enzymes in the short-term control of glycolysis. But also it is the rate limiting. This is the most important for the rate limiting enzymes in glycolysis. Okay, so these three important enzymes, the PFK1 is the most important. The PFK1, and you have to know what is the inhibitor and activator for the PFK1. They are in your original. They are in your original lectures. And the PFK1. So it can be activated by the AMP, ADP, fructose 1,6-2-phosphate, and fructose 2,6-2-phosphate, and it will inhibit by the citrate ATP in the high concentration. And remember, for this part, PFK1, you have the very, very complex regulatory modules. You don't have to remember that. It's too complex. And the third important enzyme is a fructose is in the 10th step in glycolysis. It can catalyze the PP to produce the pyruvate. And the activator is the fructose 1,6-bisphosphate, and inhibitor is ADP and adenine. But also these enzymes can be regulated by the covalent modification using the phosphorylation and the dephosphorylation. Okay, next part in this chapter is the TCA cycle. This part, what do you have to remember? You don't have to remember the every step, any steps in these cycles. But remember, after this, we are talking about is how many ATP generation. It's from the anaerobic oxidation of glucose. Help you to review. For example, if I'm asking you, it's how many molecules of ATP? It's anaerobic from the oxidation from the acetyl-CoA. It's 10 molecules of ATP. How to calculate? It's acetyl-CoA because in the TCA cycle, it have the 1, 2, 3, 1, 2, 3, the 4 NADH, and each NADH can produce 2.5 ATP. And one FADH2 can produce 1.5 ATP. And one state substrate level phosphorylation is 1 ATP here. So it's 10 ATP in total. And if I change another question, it's from the how many ATP is from the anaerobic oxidation of the one molecule of pyruvate. It's 12.5 because for the pyruvate to acetyl-CoA, it will produce a one molecule of NADH to produce 2.5, actually 2.5 ATP. And this part, if I'm asking you how many molecule of ATP from the anaerobic oxidation of the glucose, so you have combined with the glycolysis with this ATP and the NADH. Okay, so this part. Next important part is the glycogen degradation. Glycogen here. This is the glycogen synthesis from the glucose. This is the glycogen degradation here. So this step, the glycogen degradation, is firstly glycogen is going to with the glycogen phosphorylase and to produce the glucose 1-phosphate. And it will convert into the glucose 6-phosphate. And it will remove the glucose 6-phosphate. This only live in the liver, and it produces the free glucose. For the glycogen degradation, the first step, glycogen, you have to know this enzyme is very important enzyme here. Glycogen degrading enzyme is glycogen phosphorylase. It's phosphorylase here. It only cuts alpha-1,4 glycosidic bonds. Okay, it's only cuts its alpha-1,4 glycosidic bonds. But we know the glycogen, it has a branch structure. So it still have the branch structure. And it have the second enzyme. It's called debranching enzyme. How does debranching enzyme work? The big debranching enzyme will cut the alpha-1,6 glycosidic bonds. Okay, so it have these two steps here. So it firstly transfer a block of the 3 glucose residues from the end of the chain to the main, to the another chains. And this is the first, is enzyme activity. A second enzyme activity, hydrolase. Hydrolase can cut the alpha-1,4 glycosidic bonds. So its last glucose residues will cut off by this hydrolase. So the debranching enzyme has a two enzymatic activity. This is just enzymatic activities, not means two enzymes. It's just activities. So one is a transferase activity, one is a hydrolase activity. Okay. So next, from the glucose 1 phosphate, it converts to the glucose 6 phosphate. Next important part, we're talking about how does the hormone can regulate. Does the hormone can regulate carbohydrate metabolism? So firstly is insulin. Insulin is decrease. It will decrease the blood levels. Okay. So let's have a look here. This is the important part. It's glucagon. Glucagon is the important part. So firstly, you have to know what's the roles for the glucagon, what's the function for glucagon. It can stimulate the glycogen degradation in the liver, but not muscle, okay? And it induces the phosphorylation of regulated enzyme. It can raise the CMP level and activate the phosphorylase kinase. And you have to know the mechanism, the working mechanism of glucagon to regulate the glucagon degradation. Okay. Okay, so I have you review this part again. So firstly, the glucagon in the liver will combine with the glucagon receptor. You don't have to remember this part. You don't have to remember this part. Just remember glucagon combined with the receptor. And this to form the complex, this whole complex, it can stimulate and activate the PKA, the protein kinase A. This complex, including glucagon, it will stimulate and activate the PKA. And the PKA, how to activation of PKA? Because it can combine with the CMP. This is the important part in the chapter of the cellular signaling. And the activated PKA can phosphorylate the phosphorylase kinase with the phosphorylation, okay? And the phosphorylated, the phosphorylase is activated form, and to give the phosphorylation reaction to the glucagon, phosphorylase, to transfer the glucagon phosphorylase to the active form. And the phosphorylase, we know, it can help to cut the alpha-1,4 glycosidic bonds in the glucagon and help the glucagon, promote glucagon degradation. Okay, this mechanism is very important. Okay, next, you have to know the numbers here for the blood sugar. you have to know what is a normal. This is a normal range for the blood sugar. What is blood sugar? Blood sugar is what is blood sugar? Blood sugar is blood glucose level. What is this? Is a normal blood glucose level in normal people. And this is these values is for the hyperglycemia. So this is a higher with a higher blood glucose level. And this is the blood glucose level for the hypoglycemia. Okay, so you have to remember these three values here. This is the chapter 4. Next, chapter 8, nucleotide metabolism. First, you have to know the de novo biosynthesis and degradation of purine nucleotides. It have the one is de novo pathway, one is the salvage pathway. And I will tell you this part is very important. It's source of all 9 atoms of purine ring. So you have to remember all of this one. What is the source of this one? Purine ring has 2 general phases. One is generate the IMP. This is the first nucleotide generated from the purine synthesis, de novo synthesis. And next is IMP convert to the AMP and the GMP. Next part is the purine degradation. Purine is degraded to the insoluble uric acid. You don't remember these steps here, but you have to remember if you have the high levels of uric acid, you will have the gout. So you have to remember the disease here. What is the mechanisms for this disease? Because the high levels of uric levels is a high accumulation of uric acid crystals in the extremities. And you have to know what's the treatment for the here for the gout is allopurinol. Allopurinol this is a treatment. And this is the working mechanisms for allopurinol. You have to remember these working mechanisms here. This is a very important part. Okay, so next is pyrimidine de novo synthesis. It's still you have to know what is the source. What is the source for this pyrimidine ring? And the regulation of the de novo synthesis of pyrimidine nucleotide. It's for the feedback manner. The important part here is what is the difference between the bacteria and the mammals. The difference between is the king enzyme. Okay, so the king enzyme is part to regulate the king enzyme. You can go back to the lectures to review it. And this is just a general summary. Is the difference between the purine and the pyrimidine in the de novo synthesis. Okay, so this is this chapter. Next chapter is DNA synthesis. All we can say is DNA replication. Firstly, we're talking about the DNA replication. For DNA replication, you have 2 parts you have to remember. First part, generally, you have to know the every enzyme and the function of the every enzyme involved in the DNA replication. So first enzyme, helicase. Helicase is separated 2 strands of double helix. So it can break the hydrogen bonds. And so it can separate the double-stranded DNA. Next is a topoisomerase of the garase. The other name is garase. It will remove the supercolony. So the garase here, it have the 3 functional steps. Firstly, DNA garase will cut the DNA strand. It cuts just at the DNA strand backbone, not the hydrogen bonds, okay? And the second step, a DNA will reroute and remove the coins. And the third step, DNA garase will rejoin and to join the DNA strands. Next is the next enzyme, DNA polymerase 3. It's used for extension. Next is DNA polymerase 1. So DNA polymerase 1, it will to have the firstly is the exonuclease activity from the 5' to the 3'. It's used to remove the iron. What is iron nucleotide here? It removes the iron primer here. And next is the DNA synthesis. Next enzyme properties is DNA synthesis 5' to the 3'. So it will continue to get this one. And the last enzymes is DNA ligase will to seal between the two oxidation fragments. Okay. And next part, you have to remember the properties, the 4 properties of DNA replication. DNA replication is semi-conservative, semi-discontinuous, bidirectional, and the faithful. Okay, it's 4 properties. It's very important. 4 properties of the DNA replication. Next is reverse transcription. So we have the 3 enzyme activity for the reverse transcriptase. So firstly is iron-directed DNA polymerase. Use the iron as a template to get the newly synthesized DNA strand. So we'll have the DNA RNA hybrids. And the second is RNase H activity. It will degrade the iron in these hybrids, DNA RNA hybrids. The third enzyme is DNA-directed DNA polymerase. It can use this single-stranded DNA as a template to make the double-stranded DNA. Okay, so this is this chapter. Next chapter, chapter regulation of gene expression. First, you have to know some general glossary. For example, what is a constitutive expression? It's always constitutive expressed. And corresponding this, we call these genes housekeeping genes. Okay, and you have to know what is the induction, what is repression. And the properties, what it means is positive regulation, what it means is negative regulation. And very important for this part is we know the lac operin. You have to know what is the working mechanism for the lac operins. Generally, it have the 3 cases. Firstly, is only glucose is present. And the second is both lactose and glucose are present. And the third is only lactose is present. And this part, you can go back to the lecture, to this chapter lectures, and to review together. It have very, very detailed explanation. Okay, so this is the most important for my part in this semester. I'll be quick and let the second lectures going. 陈老师在吗？陈老师呢？
在的，能听见吗？
我能听见。那我这边 quit 了啊。
好的。
Okay, everyone, my part is will be finished. 陈老师，你那个投屏，
我要切一下屏，对吧？要共享一下屏。
对，你共享屏的，我真的又出去了。
哎，我看一下。哎，他人正在共享，无法发起共享。
等等，我看看啊，我结束共享，你来。
哦，那可以了吗？现在能看见吗，张老师？
可以，我能看见，那我静音了啊。
应该那就都能看见了。等一下啊，我看一下。哎，为什么有的人不能关掉他们的那个声音啊？我怎么关其他人的声音啊，郑老师？彭安佳，please quiet your voice, okay? Quiet your voice. 彭安佳。Okay, hello everyone. I will continue the class, the review class. I will give you this chapter's review and includes nucleic acid, metabolism of lipids, iron synthesis, biological oxidation, vitamins, metabolism of non-nutritive substance, and the last chapter is a cellular signal transduction. And Dr. Jung have already told you the we have 3 kinds of we have 3 kinds of questions, right? And we have single choice questions, the essay questions, and the questions of Okay, now let's start with chapter 2, nucleic acid. In this chapter, first you should remember the definition of nucleic acid. You should know that the nucleic acid is a biopolymer composed of nucleotides linked in a linear sequential order through the 3' 5' phosphodiester bonds. And in this key point, I will give you the single choice question, okay? So remember that the important bond in the nucleic acid is called the 3' 5' phosphodiester bond. And secondly, you should remember the classification of nucleic acids. In this part, you should know that nucleic acids contain DNA and RNA, and 90% of DNA are located in cell nucleus, and 10% are located in mitochondria. And also, you should know the function of DNA. It can carry and store the genetic information and determine the genotype of cells. And for the RNA, its location is mainly in cell nucleus or cytoplasm, and its function is involved in the expression of gene. And in this part, I will also give you the single choice question or the acid question. Okay. And next, for the basic unit of a nucleic acid, you should know that the basic unit is the nucleotide, okay? And the nucleic acids can be hydrolyzed into nucleotides with the help of nucleases. And the nucleotide can be further hydrolyzed into phosphate and nucleoside. And then the nucleoside can be further hydrolyzed into bases and pentose. And the bases includes purine and pyrimidine, okay? And the pentose includes the beta D ribose in RNA and beta 2' deoxyribose in RNA. And there are many differences between DNA and RNA. The first difference is the pentose difference, okay? And this slide shows us the basis. And in this part, you should remember the difference of the basis difference between DNA and RNA. In DNA, there are adenine A, G, C, and T in DNA, okay? But in RNA, there are A, G, C, U, okay? So there are 5 kinds of bases in nucleic acids, but there are the difference. Okay, there are difference between RNA and DNA. And so please remember this. And also in this part, I will give you the single choice question. Okay. And next, for the next difference between DNA and RNA is the pentose difference. In RNA, I have already told you that it contains a beta D ribose, right? But in DNA, it is the beta D 2' deoxyribose, okay? And their difference is it's on the 2' carbon atom, right? So, and the extra hydroxyl group in RNA actually can be easily oxidized by other chemicals. So this is why DNA is more stable than RNA. So this is the most important difference between DNA and RNA. Okay. And next is the definition of a ribonucleoside or deoxyribonucleoside in DNA. Okay. Ribonucleoside is in RNA and deoxyribonucleoside in DNA. So in this chapter, actually, there are many kinds of various kinds of bonds. So in this compound, we have the N-glycosidic bond. You should remember this, okay? And so, for the definition of ribonucleoside and the deoxyribonucleoside, they are the compound which 2 kinds of pentose linked with bases respectively by the N-glycosidic bond. And this compound is called the nucleoside. Okay, this compound is nucleoside. And for next definition is about the ribonucleotide and in RNA and the deoxyribonucleotide in DNA. And for the in this compound, it is a nucleoside connect with the phosphate group, right? So the nucleotide is a nucleoside containing a phosphate group, and the connecting bond is called the ester bond. Okay, the connecting bond is the ester bond. So you should have a clear you should have a clear review about the ester bond, the N-glycosidic bond. Okay, remember this. And next is the definition of nucleic acid in detail. And nucleic acids are lots of nucleotides are joined together to form a long polymer chain by covalent bonding between the phosphates and pentose. And for any one nucleotide, the phosphate attached to the hydroxyl group at the 5' position of the pentose is in turn bonded to the hydroxyl group on the 3' carbon of the pentose. So remember the definition of nucleic acids in detail, okay? And the next important bond here is the 3' 5' phosphatidyester bond. And here, this one, actually the 3' 5' phosphatidyester bond, it is not a traditionally bond. It is a group linked to nucleotides, and we show it in this gray frame. In this gray frame. And since each phosphate hydroxyl group is an ester bond, the linkage between the two nucleotides is called the 3' 5' phosphatidyester bond. So you should have a clear view about this bond and other bonds. Okay. And next is about the primary structure of nucleic acids. The primary structure of nucleic acids is the sequence of nucleotides. Okay, nucleotides, and the difference between bases are the main difference between nucleotides, and this is also named as the sequence of bases, 碱基序列. Okay. So this point is also, I will give you the single choice question. Okay. And next one is about the DNA secondary structure. You should remember that it is a double helix structure. It is a double helix structure. And about the double helix structure, you should know the Chekhov's rule. The Chekhov's rule. And there are 4 key points. The first is that different species and different individuals have different base composition in DNA. And the second is that the different organs or tissues in one individual have the same base composition in DNA. And the third one is the base composition in DNA does not change with age, nutritional status, and environment in particular tissue. And for the last one, you should remember clearly, for a certain individual, the concentration of A equals to concentration of T, J equals to C, also A plus J equals to J plus T plus C. Okay, so remember this one clearly. Okay, and I will give you some single choice questions here. And for the next structure of DNA, it's an advanced structure. There are some kinds of advanced structures. The superhelix, or we also call it the supercoil, 超多旋结构. And remember the definition of superhelix or supercoil: adding or removing twists in DNA may induce the DNA supercoiling. Okay. And next one, there are 2 kinds of superhelix: positive supercoil and the negative supercoil. Their differences are that in positive supercoil, it is adding some twist to a typical relaxed DNA double helix. And for the negative supercoil, it is removing the twist in DNA, causes a negative supercoiling. Okay. And for the eukaryotes, the DNA can be packaged in chromatin with various proteins in eukaryotic cells. As a major proteins of chromatin are known as histones. And the basic unit for the eukaryotic chromatin is the nucleosomes. And this picture shows us the composition of nucleosomes. It contains the DNA and histones, and the DNA is about 200 base pairs. And for the histones, there are four kinds of histones. They are H1, H2A, H2B, H3, and A4. Okay. So please remember that the nucleosome is the basic unit of the chromatin, and the nucleosome contains DNA and histones. Okay. And this slide shows us the levels of chromatin structure, and you should know that the DNA is the most basic unit for the chromatin, and the DNA can be packaged to form the nucleosome, DNA, and histones, right? And then the various nucleosomes can form the condensed fiber about 30 nm, and then it can be condensed to the chromosomal loops about 300 nm, and then to the chromatin, and then last to the chromosome. Okay, and so this is the levels of a chromatin structure. And also, I may give you some single choice questions in this part. Okay. And next, for the classification of RNA, there are many kinds of RNAs, includes mRNA, messenger RNA, tRNA, RNA, and other non-coding RNAs. And you don't have to remember all kinds of non-coding RNAs in this part, but you should remember the heterogeneous nuclear RNA. It is also can be written short as the hnRNA. And you should know that it is the pre-mRNA. Okay, it is the eukaryotic pre-mRNA, and also it is pre-mRNA in eukaryotic organisms. Okay, so remember that hnRNA it is located in nucleus, and its function is as the eukaryotic organisms pre-mRNA. Remember this, okay? I will give you some single choices, single choice questions about this part. And next, a single choice questions I will give you is about the RNA. The proteins are synthesized in ribosomes, which are composed of RNA. Okay, and the ribosomal RNA is the most abundant RNA in cells. And for the composition of RNA, it contains the RNA and the protein. It can form the ribosome. Okay, it can form the ribosome. And for the function of RNA, it is first, it is the most abundant form in living cells, and also it is part of the protein synthesis apparatus. And also the RNA is a catalytic component of the ribosome. So in this part also, the key point in this part is ribosome 和酶. It is the only RNA which has catalytic functions. So remember that the ribosome can catalyze specific RNA degradation, and it can be also functions in RNA splicing modification. Okay. And next one is the DNA denaturation. For the definition of DNA denaturation, you should remember it. You should remember it clearly. Okay, you should remember this definition clearly. And it is some physical and chemical factors can lead to the breaking of hydrogen bonds between DNA double-stranded complementary bases and dissociation of DNA double strands into the single strand, okay, into single strand. And this picture shows us the DNA denaturation. And next chapter is metabolism of lipase. And in this chapter, firstly, you should also remember the definition of a lipase. Lipids are a class of macromolecules with various and complex structures. Fat and lipoid are called lipids, okay? And you should remember the lipids can be classified into fat and lipoid. And fat is also named as a triacylglycerol, TAG. Okay, TAG. TAG also can be written as triglyceride, TG, 甘油三酯. And lipoid includes cholesterol, cholesterol ester, phospholipids, glycolipids, and sphingolipids. Okay, so remember that the lipids can be classified into fat and lipoids. Okay, and remember the examples of the lipoids. And next, the key points is about the triglyceride. It is fatty acid esters of glycerol. Okay, it is a fatty acid esters of glycerol. So for more detail, you can have a review about the PowerPoint we talked about in our class in the chapter 5. Okay, and next is fatty acids. Fatty acids, in this part, you should remember the classification for fatty acids. It contains the saturated fatty acids and the unsaturated fatty acids. The difference between saturated and unsaturated fatty acids is that the saturated fatty acid don't have any double bonds, okay? And the unsaturated ones contain at least one double bond. And for the unsaturated fatty acids, it contains the monounsaturated fatty acids and the polyunsaturated fatty acids. Okay. And next key point about the fatty acids is essential fatty acids. In this part, I will give you the single choice questions, and you should remember the components of the essential fatty acids. There are three kinds of essential fatty acids. They are linoleic acid, linoleic acid, and the arachidonic acid. Okay, you should remember these three examples of the essential fatty acid, and there may be a single choice question for you guys. And next one is about the synthesized unsaturated fatty acid derivatives. Also, I will give you the single choice questions of these key points, okay? And for the next section is about the fatty acid catabolism. And this part is the most important. It is the most important part of the whole chapter, okay? It is the most important part of this chapter. And there may be some acid questions for you guys, okay? And most importantly is I will give you a review about the beta oxidation of fatty acids, okay? And firstly, you should remember the definition about the fat mobilization, okay? And you should remember the definition of fat mobilization. It refers to the gradual hydrolysis and release of free fatty acids and glycerol from fats stored in adipocytes for oxidation by other tissues under the action of fatty lipase, okay? And remember the key enzyme in this process, okay? And secondly, the glycerol is converted into glycerol 3-phosphate and used in carbohydrate metabolism. And most importantly, you should remember the process of beta oxidation of fatty acids. Okay, and the beta oxidation is the core process for fatty acids catabolism. And the first thing you should remember, it happens in liver and muscle, and in cells, it happens in cytosol and mitochondria. And it contains 3 main processes. The first process is the activation of fatty acids. You should remember the process here and the K enzyme here. Okay, the K enzyme you should remember very clearly. Okay, and also in this process you should remember the materials, the K enzymes, and the products. Okay, and for the second process of the beta oxidation, you should remember it is the transport of acyl coenzyme A to the mitochondrial matrix via carnitine. Okay, and for the third process, or the third process of the beta oxidation, it is the oxidation of acyl coenzyme A to acetyl coenzyme A, FADH2 and ANDH. Okay. 多少是在吗？多少是在吗？我后面会讲的稍微简单一点，但是我要拖一会儿，10分钟可以吗？
可以。
10分钟啊。sorry，不好意思。
没事，好。
有点多，讲太多了。
好。
OK，好，那我继续啊。I will continue. Okay. And in the third process of beta oxidation, you should remember the oxidation of acyl coenzyme A to acetyl coenzyme A, FADH2 and NADH. And in this process, you should also remember all materials in each processes, and also you should remember all the enzymes in this process. Okay, and you should know they are the hydrogenate process, hydration process, dehydrogenate process, and the last process is silysis. Okay, and this is the most important part in this chapter. And next key point in this chapter is about the ketone bodies. Okay, ketone bodies, you should remember the components of the ketone bodies. They are acetoacetate, beta hydroxybutyrate, and acetal. Okay, and you should remember their synthesized location in the mitochondria, in hepatocytes, and it can be utilized in mitochondria in extrahepatic tissues. And you should remember the difference between the synthesized location and the utilized sensation. And you should remember it very clearly. Okay. And next is the metabolism of a lipoprotein. In this part, you should remember it can be classified into the CM, VLDL, LDL, and HDL by the ultracentrifugation. Okay, ultracentrifugation. And in this part, you should remember the different products of different lipoprotein transports. You should remember that CM transports the exogenous triacylglycerol, VLDL transports endogenous triacylglycerol, and LDL transports the cholesterol from liver to other organs. HDL transports cholesterol from other organs to liver. Please remember this summary very clearly. Okay. And next chapter, I will introduce the ion synthesis. In this chapter, first you should know that there are two ways of ion synthesis. One is the transcription, and the other is the ion replication. Okay, and you should remember the definition of transcription. It is the process of ion synthesis using the DNA as a template. Okay. And next, in the section 1, you should remember the templates and enzymes in prokaryotic transcription. Okay, and in this part, you should remember the ion polymerase very clearly. Okay, and the difference between core enzyme and the hollow enzyme of ion polymerase is that in hollow enzyme there is a more sigma subunit in hollow enzyme. Okay, and this sigma subunit is mainly involved in transcription initiation stage. Okay, and this is a difference between hollow enzymes and core enzyme. Okay. And for section 3, you should remember that in eukaryotic transcription, they are more complex than prokaryotic transcription. There are the transcriptional factors, or we can also call it the transacting factors involved in the transcription. And also, you should remember the cis-acting elements in eukaryotic transcription. And there are many kinds of cis-acting elements here. You should remember the enhancers, the transcription start sites, the translation start sites, the introns, and exons, etc. And you should remember that the difference between cis-acting elements and transcriptional factor transcription factors. Remember this clearly, okay? And next one, you should remember the definition about the exon and the introns, okay? Remember the definition of exons and introns. And next chapter, chapter 12, is about the biological oxidation, 生物氧化. In this chapter, first you should remember the definition of the oxidative respiratory chain. And next, you should remember the 4 kinds of complexes of human mitochondrial respiratory chain complexes. You should remember in this table that the enzyme name of each complex and the binding site of each complex. Okay, remember this. And I will give you some single choice questions of this chapter, of this table. Okay, and next important part in this chapter is the inhibitors in respiratory chain. And also, I will give you some single choice questions in this part. You should remember each inhibitor of complex 1, 2, 3, and 4 very clearly, okay? And next part is the metabolism of non-nutritive substances. And in this part, you should first remember the definition of biotransformation, okay? And you should know the classification of non-nutritive substances. And secondly, you should remember the biotransformation site very clearly. Liver is the most important organ of biotransformation. And next one, you should remember the biosize, the 2 kinds of biosize, and the bioacids. Okay, the bioacids, you should remember the classification of bioacids, and also you should remember each example for each bile acid. You should remember the cholic acid and the desoxycholic acid. They are examples of free bile acids. And for the conjugated bile acids, they are taurocolic acid and glycolic acid. You should remember these examples very clearly, okay? And I will give you the single choice questions here. And also another kind of classification of bile acids, it can be classified by source. It can be classified into the primary bile acid and secondary bile acid. Also, you should remember these examples very clearly. And next section, you should remember the materials of heme biosynthesis, okay? And for chapter 13, vitamins, first you should remember the classifications of vitamin, the lipid-soluble vitamin and water-soluble vitamin. Remember the classification of each kinds of vitamin, okay? With A, D, E, and K, they are the lipid-soluble vitamins. And remember the disease caused by lack or excessive intake of vitamin A, and lack intake of vitamin D. Remember the diseases very clearly, okay? Remember it clearly. And for the water-soluble vitamin, you should also remember the classification. It contains B and C vitamin, okay? And also, you should remember some disease examples for the deficiency of vitamins, okay? And for the last chapter, I should introduce you the receptors and the ligands. And you should remember some examples of the intracellular receptor and the cell surface receptors very clearly. I will give you some single choice questions in this part, okay? So, and also you should remember the interactions between receptors and ligands. They have some common characteristics. They have the highly specific, highly specificity, highly affinity, and the saturability, reversibility, and some specific mode of action. Remember these kinds of common characteristics between receptors and ligands clearly, okay? And also, I will give you some single choice questions here. Okay, this is up close. And next, 葛老师，
我在。
好了，我结束了，
你结束了是吧
这边？退出了。
哦，好。
我要结束共享先。
फिर बतिया रही है अपने घर वालों से।
好了。
पढ़ा लो यार।
अरे क्लास का ये सर है ना बोल रहा है वो बोलने लगी
था। हेलो
Hi, can you hear me? Uh,
你讲吧。
听到是吧？听到是吧？
听得到，你讲吧。
好。We will go through the chapter 1, the structure and the functions of the protein. First, what is the protein? You need to know, and we have the biological importance of the protein. So all this I have explained in the class, so you need to know this is important.